Welcome to Bio 212, Anatomy and Physiology 2, and we are working on Chapter 21, where inside the capillary system, delivery system, we are now getting fluid to capillary beds, and what we're talking about is hemodynamics. So what are the factors that are going to change blood flow through arteries to get to a capillary bed that will affect diffusion or perfusion of those tissues, right? So and that's what Mother Nature actually has to think about all the time. She's thinking, just because I'm getting blood there doesn't mean it's the most effective way to get blood there. Okay, so blood flow is that volume of blood that flows through any tissue in a given period of time in milliliters per minute. Big deal. Okay, total blood flow is, of course, obviously cardiac output, and cardiac output, obviously, is heart rate times stroke volume. Okay? The distribution of cardiac output depends on the pressure differences that drive blood through the tissue. Blood flows from areas of higher pressure to lower pressure. So that means a capillary bed represents a field of lower pressure. Okay? And the higher the resistance that it encounters, the lower or the smaller the blood flow that actually moves into the space itself. And let's take a look at what this looks like inside your body. Okay, so this is systolic and diastolic blood pressures when you actually, so these are the heartbeats you were listening to. Now what we're going to do is we'll put a pressure cuff on some of you guys inside class after you've read the book in this section about it, right? And you're going to see that 120 over roughly 75, 80 here, this is what you're going to collect as your systolic and diastolic volumes. And this is inside the aorta as it passes down through the arteries, collecting the pressure inside your arm. You're going to see that the actual pressure is dropping. When it hits the capillary bed, it should be somewhere around 36, right? That makes sense. We showed you a minute ago. And as it exits, it should be somewhere right around 10 or something like that, right? But notice as it actually approaches the right atria, it is now at 0. So everything I've told you fits in with what we actually see when you're looking inside the body. So when we're thinking about what it's running into, we call this resistance, and that depends on the size of the lumen. Okay? What happens... It doesn't... Oh my God. Sorry about this, guys. Okay. Okay, vasoconstriction obviously makes the lumen smaller, meaning it has greater resistance, and therefore the blood viscosity could actually have the same thing. The more dehydrated you are, the ratio of cells, formed elements, and plasma protein makes that more thick, and therefore it's harder to push. Okay, total blood vessel length is the resistance directly proportional to the length of the vessel. So your textbook puts this in there, and I don't really know what I think about it, to be quite honest with you. I'm not quite sure what their goal is, other than there's a series of angiogenesis steps that occurs to support adipose tissue inside your body. And for every 2.2 pounds of adipose tissue you have, you've added about 400 miles of blood vessels to support that. And thinking about that in terms of physiology, and that's the only way I want you to think about it, it would be, well, what is the extra work the heart has to do to push that blood? Okay, that extra 400 miles. Remember, 400 miles relative to what's inside of your body right now. Put that in there, right? Inside your body right now, as an adult, you have somewhere around 100,000, 90 to 100,000 miles. So 400 miles is insignificant relative to that. But as you age, you can imagine the ability of the heart to push additional mileage just becomes harder and harder, more of a stress. Okay? Venous return, obviously, I've already told this to you. We have to get to 0 millimeters pressure, okay, to actually get to the right ventricle. So how does it do this? Well, we use two other pumps. We use the skeletal muscle pump. Okay, that means, if you've ever heard the saying, to stop is to die, well, that's what we mean, okay? Because if you're not moving, your skeletal muscle pump isn't working well, nor is your respiratory pump, meaning your inhalations and exhalations, okay, also drive blood movement inside of your body. Think about some of the exercises we tried inside class, okay? And what's going to happen, of course, is as the large muscle groups move, it increases the tone of the actual vessels themselves, such that at the end here, you can see a map that makes sure you increase mean atrial pressure. Now, don't memorize this figure. That's not what's important. This is a review for what you saw inside chapter 20. Okay, and this is important, and I really do mean that. I mean, if I'm you and I'm thinking, oh, how am I going to study for all this material? This is one really good way to do it. Okay, because each of these increases heart rate, each of these increases stroke volume, therefore you change cardiac output, but you also have to take into consideration the increased systemic vascular resistance to now measure the mean atrial pressure inside of the body. And in doing that, you've just looked at the entire organism, which is pretty amazing. So how fast is it moving? Well, it's not a physics class. So first of all, let's not worry about how it's actually, what its speed is. But remember, I told you it takes normally about 1 minute at rest, okay, to get blood from your right atria back to your right atria. Okay, that's pretty amazing. So, whoops, where'd you go there? Okay, there's a relationship velocity of blood flow and total cross-distance area, cross-sectional area. Okay, and notice what's going to happen here is cross-sectional area increases as you go into a capillary bed. This is how you get diffusion to occur. You have more tributaries for the fluid to move through, and the pressure is dropping, so it allows it to move out easily, which is an engineering feat in and of itself. Okay, and that controlled blood pressure, of course, and blood flow is what allows you to maintain the actual role of diffusion. Notice I keep saying diffusion and switch back and forth between perfusion. Remember, on your test, we'll be talking about total peripheral resistance as it relates to the filtration and reabsorption that occurs inside of a capillary bed. Okay, and each of these will change that. Why don't I type that in there for you? Relationship of, that doesn't spell relationship, whatever. Relationship of total peripheral resistance, TPR, okay, to filtration, T-R-A-T-I-O-N, and reabsorption at cap bed. Okay, that is important. Without it, it doesn't work. The whole thing doesn't work. Where cap bed means capillary bed here. Okay. Well, of course, we're going to have to interpret this, and fine well what's going to be interpreting it. It's going to be the cardiovascular center, which we were introduced to in Chapter 20. Okay, it's inside the medulla. It regulates heart rate and stroke volume, and it controls neural, hormonal, as well as local negative feedback systems that regulate blood pressure. Okay. Together, cardiostimulatory and cardioinhibitory centers inside of it regulate all of the outputs as you collect materials, both higher brain regions as those sensory receptors. And you saw those last chapter, right? So now let's add on some detail. And there's only one difference inside this figure than the previous figure, right? Take a look at what's actually being measured here. Monitoring. Hydrogen protons, carbon dioxide, and remember I said oxygen, but it's less important to us. Notice how they're describing proprioceptors, joint movements, blood movement, stuff like that. Now down here, you're going to see that there are vasomotor nerves from the sympathetic portion of the cardiovascular center that regulate vasoconstriction. And this is really important. What I mean by that is you're seeing that there is no parasympathetic, for vasodilation. Okay, and that's not partially true, but what you're seeing here is from the cardiovascular center. Use vasomotor nerves to regulate sympathetic tone of the smooth muscle for all of the blood vessels. Okay, we're thinking primarily about arteries, though. Look at that. I said, don't talk about veins, and there's the first word that came out of my mouth. Okay, cool. So how do we do this? Baroreceptors, okay? These are pressure-sensitive receptors inside the internal carotid arteries, okay, and other large arteries, internal carotids and other neck and chest. Boom, boom. Carotid sinus reflex regulates blood pressure inside the brain. The aortic reflex regulates blood pressure inside the systemic circulatory system. Okay? When blood pressure falls, baroreceptors are stretched less, and therefore you slower the impulses to the CV, and that is going to decrease parasympathetic stimulation. That should make sense, I hope, because you have to increase heart rate. Okay? Chemoreceptors, okay, these actually are also inside the carotid bodies in the aortic arch, detect low O2, hypoxia. But notice before this, anytime we actually made red blood cells, hypoxia was detected inside the kidneys. Hypercapnia, which is excess carbon dioxide, and acidosis are going to signal to the cardiovascular center. Okay, and then we're going to adjust. Okay, not only going to adjust the heart rate and blood pressure, but you also adjust the respiratory rate. Because now we have to make this work in coincidence, right? You're running to hit the ball on the pitch. Okay? Because you're running, you need more oxygen. You need to get rid of more carbon dioxide. Okay? And that means you need to coordinate all of this. Okay, so here's a fairly decent image of how this all works. And I do want you to pay attention to this, because here's the baroreceptors in the arch of the aorta, baroreceptors inside the carotid sinus. Notice we've identified the glossopharyngeal nerve as taking in sensory materials to the cardiovascular center, and you can see that you actually have the vagus nerve collecting its type of information. Also, at the same time, it has motor responsibilities, SA node, AV node, and now take a look at this. This is the CAN, cardiac accelerator nerve, and it changes ventricular myocardium, both right and left. Okay? Cool. So with all of that in place, you can work your way through this negative feedback system. As blood pressures increased, well, guess what? We shut off the increased heart rate that actually has to happen as a result of that. Okay? And here's something that you're going to notice. Okay, way down here at the bottom. We've spoken about cardiac output. Think about vasomotor output as it relates to increasing systemic vascular resistance. To maintain the efficacy at the capillary beds, sometimes you actually have to increase pressure. And that should make sense. If blood pressure is falling too low, you know fine well things aren't going to work all that well. Well, how do we do this hormonally? And lo and behold, here is our old friend, the RAA pathway, where renin is released by the kidneys and blood volume falls or below blood flow decreases. Angiotensinogen is going to be converted into angiotensin 1, and inside the heart we use the lungs we use ACE on that to make angiotensin 2. And remember, from your first exam, you should remember the angiotensin 2 is a potent vasoconstrictor. Okay, the aldosterone will save salt inside the body, which saves water inside the body, which raises blood volume, and therefore pressure. Okay, epinephrine and norepinephrine, they actually cause both cardiac output increase, but also they're going to act on total systemic resistance. Atrial antidiuretic hormone, or vasopressin, okay, is also going to increase blood volume because it's going to stop you from releasing water. And now we're adding on to our list, okay? And our list means we're going to be adding on another hormone. And this is a new one. It's called ANP, atrial natriuretic peptide. It's released by the cells of the atria of your heart. Okay, low blood pressure, okay, will cause vasodilation and therefore loss of salt and water inside the urine. Its goal is to reduce blood volume. And I don't like the way, let me rephrase that. A way to think about it is that if there's too much pressure inside the atria, it means the effectiveness of the ventricles is going to be diminished over time. So it sends a signal using A and P to the kidney to say, look, get rid of some salt, get rid of some water. By decreasing the volume inside of blood, we can now increase the efficacy at capillary beds. And that, I think, is probably the most helpful way to think about what ANP actually does. Now, At the end of the day, this is all auto-regulated. You don't have to think about any of this, right? Okay, because it's going to match metabolic demands. It has to do this. And the more you exercise, the more you realize it's easier to stay out exercising. But if you take a couple weeks off, okay, metabolic demands have dropped, and therefore autoregulation is going to drop. What's going to happen is you actually have two types of stimuli. You can have temperature changes and myogenic responses. Go jump in an ice cold ocean. You'll find out what I mean about that. And what's going to happen, of course, is they result in vasodilating and vasoconstricting results as a result of the chemicals that are put inside your bloodstream, epinephrine and norepinephrine versus, say, acetylcholine. So at the end of the day, let's draw this one more time. Okay, let's go blue first. Fantastic. What we've done is we've built vessels, right? And those vessels are responsible for getting materials to capillary beds. And these capillary beds have to be controlled such that the rate of perfusion into these tissues makes it effective for all of the cells inside of this area. Let's go over here with this, right? So we're not, I mean, even though we're talking about systemic resistance changing filtration and reabsorption, so let's go F and R, F, R, where, oh, come on, put the R down here, there we go. Okay, so filtration and reabsorption, right? You have to keep in mind that the heart itself, so let's put this way the hell over here. I'm going to put, this is the left part of the heart, right? That's the left part of the heart, and you know that because we're going to put in just a really small space here for that. Put that over there. Okay. And this is the right side of the heart, where blood's going to be ultimately returned to at 0 millimeters of pressure. Okay, I need to make the ventricle here just a little bit larger, but not as large as the others, right? And you can begin to see that anything we do to change heart function changes diffusion or perfusion rates here. Remember, extra interstitial fluid inside of this space gets collected by the lymphatic system. So we actually pull that out of there. And we've actually also added to our repertoire of hormones. Notice how many times I mentioned epinephrine and norepinephrine here this evening, okay, or this afternoon. Okay, we indirectly mentioned acetylcholine again because we were talking about the parasympathetic nervous system. We went over our RAA pathway again. Okay, we went over ADH. Okay, and then we also threw in a new one. That's a D and that's an H. Okay, we threw A and P. Okay, where you had too much fluid inside the body, so it's from the atria, sends a signal to your kidneys so you can remove volume from the body and increase the efficacy at the circulatory system. Because remember, at the end of the day, it's these cells here that have metabolic needs that your circulatory system is supporting. Okay, that's a pretty good place to stop. That will complete chapter 21.